Okay, so today we are moving on with our discussion, getting into the adaptive immune system. Okay, but to start off with, I wanted to finish up with a couple slides that we did not get around to discussing at the end of last lecture. Remember, we were talking last time about natural killer cells and how natural killer cells are these special types of lymphocytes, large lymphocytes, that actually come from, if we were to look at the cartoon of the lineages of the innate effector cells and adaptive effector cells, the natural killer cells actually come from the adaptive side, but yet they function more as innate effectors. And later on, when we talk about the evolution of how our immune system sort of came to be set up the way it is, it'll make more sense to you why there's that kind of funny overlap. So let's first start off by wrapping up a couple important things about natural killer cells. All right. Remember that natural killer cells work by not directly attacking pathogens and eating them up like macrophages do or neutrophils do, right? Those are sort of the classic innate effector cells that target pathogen. Right? Natural killer cells, they're more concerned with identifying the cells of our body that are infected with pathogen or might otherwise have some other evidence of disease. And then the natural killer cell needs to decide whether to tell that target cell to die. So in the case of viral infection, that's what we looked at last time. If you have a virus infected cell that's lost control of itself and it's making virus particles, right? I mean, that cell's kind of a goner anyways, but it can cause a lot of trouble if it releases tens of thousands of virus particles. So a natural killer cell can synapse with it, meaning it presses up against that cell, right? And through interactions of proteins and receptors on the surfaces of the two cells, the natural killer cell can tell that infected cell to undergo apoptosis, or just basically self-destruct and take itself out of the picture. So it's no longer going to be this virus factory spreading viruses in you. Right? So an important thing to think about is that we've talked about how many cells respond to cytokine signals, the small little proteins that are released by cells as a way to communicate with each other. We saw how, for example, cytokines are a way for one type of immune cell to attract another immune cell. We also learned about special cytokines that are involved in inflammation that causes certain physiological changes in the different tissues of your body, like giving you fever or getting your liver to make special proteins that help out with complement protein fixation. But we haven't yet talked about how two different cell types can communicate back and forth with each other. So that's a good way to think about this set of cartoons here. Here we have a natural killer cell that is floating around in an inactive, not activated state. Right? So natural killer cells, they're kind of very powerful killing cells. They can go around, bump into other cells and tell those other cells to die. So as a result, natural killer cells normally are not activated, right? Because it's kind of dangerous to have them just kind of going around, potentially mistakenly killing other cells. So here we have an inactive natural killer cell. Here we have a macrophage inside of infected tissue, and the macrophage is busy eating up or phagocytosing virus particles, right? So that's what these little things are here. And you can see the macrophage is doing a pretty decent job. It's eating like one or two viruses, and as a result, the macrophage begins to send out cytokine signals. So these cytokine signals consist of, for example, CXCL8 or IL-8. So remember, the main sort of cell that will respond to IL-8 that we learned about are the neutrophils that are released by your bone marrow, and they're circulating through your blood, and they'll arrive at a certain spot inside of your tissues where the blood takes them, and they stop there, and they kind of sniff out or smell the IL-8 that's being released by the infected tissue from the macrophages, and that gets the neutrophils to go into the tissue and help out with the fight. Right, so we said that was a good example of one immune cell type that responds to IL-8. So natural killer cells also respond to IL-8. So like neutrophils, they would have this dark green receptor on their surface, which is a CXCL8 or IL-8 receptor, right? That's like the nose that sniffs out the the cytokine signal. And so that will also recruit natural killer cells to see what's going on down here. The macrophage also sends out this cytokine interleukin 12 or IL-12, right? So the natural killer cell has a light green receptor protein on its surface that binds or detects this light green IL-12. And that makes the inactive natural killer cell, it's like curious, right? So it presses up against the macrophage and they communicate with each other. So different signals in the form of proteins that are on the surface of the macrophage are received by different receptors on the NK cell. And the IL-12, importantly, will activate the natural killer cell. So the natural killer cell then will begin to multiply. So from one, ends up in this cartoon, there's 4 of them. And importantly, the natural killer cell begins to prepare all of these little vesicles inside of it that are filled with those terrible sort of molecular message signals that can be used then to make another cell die, right, to kill itself. So then many of these activated natural killer cells then, they will now float away and start looking for infected cells that show evidence of this virus being infected inside the cell. But the NK cells do something else. And this is what I mean when the cells talk to each other back and forth. The natural killer cell starts expressing interferon, right? In particular, the type 2 interferon or interferon gamma, right? So only natural killer cells express interferon gamma. Or I should say they are among a very limited number of immune cells that express interferon gamma. And interferon gamma is the natural killer cell responding or talking back to the macrophage. So you can see here, this natural killer cell stuck around here and is sending interferon gamma. And the macrophage has the orange receptor for interferon gamma. That's what triggers the macrophage to kind of really start ramping up its activity. So in a way, this is like a conversation, right? The macrophage on the left says, hey, natural killer cells, come here. I have something to kind of tell you about, right? The natural killer cells come, right? They get the message, they activate, and in return, the natural killer cells are saying to the macrophage, hey, you're doing a good job, but it's really better if you start ramping up your activity. So as a result, the macrophage, you see, increases the rate of phagocytosing the bacteria. So now it's eating like 6 of them at the same time or something, whatever this cartoon is showing. And it also increases its production of cytokines. So it's a conversation back and forth mediated by these cytokine messages. Right. Another type of innate immune cell that also plays a role in the adaptive immune system are these dendritic cells. So there's different types of dendritic cells or DCs. The one that we will first start talking about, and this is the common form that most people think of when they think of dendritic cells, is what we call a conventional dendritic cell. Right? So this is a dendritic cell here. Right? A dendritic cell samples the environment. It's kind of in there, in the tissues, crawling around with the macrophages, and it's constantly picking up scraps of things, bits and pieces of dead cells. These might be pieces that come off of pathogens as well. It also phagocytoses things. So virus particles, bacteria, the dendritic cell will also eat them up, right? But its main job is not to eliminate things like bacteria and viruses directly, because it has very limited ability to do so. Its main job is to get all these little samples of things that are happening and bring those samples to where other immune cells are hanging out, ready to receive these evidence that something is going on. So I kind of think of dendritic cells as kind of like, like your pet dog or pet cat who goes off in the morning, is running around the neighborhood, whatever, and then they bring you back something like a dead rat, right? And they present it to you because they love you. They're like, yeah, I love you. Here's a dead rat. And you're like, oh, thanks, right? Dendritic cells are like that. They pick up on all kinds of things and bring them back to present to other immune cells, right? And so here, this cartoon over here shows that In the tissues, they are there with the macrophages already, and you can see the dendritic cells are collecting all these little red dots, and these red dots represent scraps, bits and pieces of bacteria. These dendritic cells then drain into these special vessels that we will study later. This is part of your lymphatic system, and the dendritic cells bring that evidence that it found, all these bits and pieces of things, to places like lymph nodes. That's what this sort of jelly bean shaped thing is, which we'll study quite a bit later on. It's there that you have a whole bunch of other immune cells hanging out. And those immune cells then have an opportunity to actually see this evidence of infection, right? That they otherwise would not see, because this could physically be some distance away from where the infection is actually taking place. Okay. So dendritic cells are kind of like these little lovable, cells that just kind of like collect stuff and show things off for other cells. But it's another example of how two cell types can talk together. So let's look at this here. Important interactions take place between these dendritic cells and natural killer cells, right? As another way for natural killer cells to kind of see that there's evidence of something going on that they should get excited about, right? Here are three different sort of scenarios. Let's talk about this here, right? On the left is kind of your sort of very generic, canonical, simple way of thinking about this. You have a dendritic cell here that's carrying on its surface all these little bits of, let's say these red dots are pieces of the bacteria, right? And you have this inactive natural killer cell that's minding in its own business floating by, right? When they synapse, the natural killer cell has a chance to see these red dots, right? This is the evidence that the dendritic cell brought from the site of infection, right? And that can help activate the natural killer cell. Right, so then the natural killer cell begins to activate. It starts making all those little vesicles filled with horrible cytotoxic substances, right? And then they will go off and try to find, cells in the body that might be where this pathogen has infected. Right. So the middle panel and the right panel are two other scenarios that happen in real life that have to do with the relative number of dendritic cells versus activated natural killer cells. In this middle panel here, let's pretend that in this case, here comes a dendritic cell. It's carrying all those little red dots, little scraps, bits and pieces of the bacteria or the virus, right, into something like a lymph node, where there's already, in this case, a whole bunch of already activated natural killer cells, right? So these natural killer cells, they're already, working, right? If it turns out that the number of those activated natural killer cells outnumber the number of dendritic cells that are bringing in more pathogen, kind of an interesting thing happens here. The natural killer cells actually tell the dendritic cell to die, all right? So the dendritic cell actually becomes a target of the natural killer cell's cytotoxic messages, right? Why? Because in doing so, what the natural killer cells are doing is they're suppressing the dendritic cell from further moving on and potentially doing something like waking up the adaptive immune system. And you're like, why would they want to do that, right? Because I thought the whole point is eventually you want to wake up your adaptive immune system. No, remember, your body wants to hold off on activating your adaptive immune system until your innate immune system has really had a chance to see if it can eliminate the pathogen, right? Before you really start with the expensive stuff like the adaptive immune system. So because you already have a lot of activated natural killer cells in this scenario here, this probably means your immune system is already well on its way to defeating the infection. Right? So in that case, the natural killer cells don't want the dendritic cell to take this any further. Right? But the opposite would be the third panel here. If you have relatively few already activated natural killer cells and a lot of dendritic cells coming in carrying evidence of an infection, right? This probably means, right, that the infection is kind of bad and your immune system hasn't yet kind of gained the upper hand. So in that case, right, the natural killer cells will actually further activate or stimulate the dendritic cells and tell them through cytokine messages to go on and start waking up the adaptive immune system, right? Because you might be dealing with a lot of trouble here. All right, this is it. We got to get the adaptive immune system moving. All right. And here is a nice image here. This is a dendritic cell, right? And so, by the way, dendritic cells, they change their shape quite a bit. But one of the morphologies that's very typical is this very messy looking, highly ruffled kind of cell morphology. And that's because all these little ruffles are places where bits and pieces and scraps of antigens are actually being presented by this dendritic cell. And here it is communicating with something like a natural killer cell, some kind of lymphocyte, right? So it's interesting to think, I wonder what kind of discussion they're having here. Okay, so that was the last couple of slides from last lecture that I thought was important for you to be aware of. So let's move on and really talk about the adaptive immune system now, right? Because the previous, whatever it is, 6 lectures that we've had this semester is all about the innate system. I said, let's get the innate system out of the way because it's relatively straightforward, right? Complement proteins, macrophages, some cytokines, and then you have some things that are a little bit different, like the natural killer cells and the dendritic cells, but that's kind of it, right? Let's move on to the adaptive immune system. To start off with, let's discuss the context of why, the fact that we have an adaptive immune system not only is crucial for our survival, but surprisingly is not actually the most common sort of arrangement that life on Earth depends on. Most of life on Earth depends simply on an innate immune system to deal with things, right? Because most life on Earth are not as complex and as large and as long-living organisms as we are, right? So back when life started, you had bacteria 3.5 billion years ago. So even lowly little bacteria had to have ways of identifying threats like viruses, right? Like phage, lambda phage viruses that attack bacteria. So that's why we learned that they already had mechanisms to detect viral genomes, and that got carried in to eukaryotic cells in the form of mitochondria, right, that are still involved in helping our cells detect viral infections. Then about 2 billion years ago, you have eukaryotes that began to develop, right? And importantly, eukaryotes had to develop certain receptor proteins on their surface that could distinguish between self versus non-self. What do I mean by that? Really quick, think about this amoeba over here, right? An amoeba is about as simple of a eukaryote as you can get, but it's already way more advanced than something like a bacteria. So here's this amoeba that's eating something, right? This little scrap of who knows what this is, but the amoeba wants it. And so it encircles it and phagocytoses it, right? How does it know when it reaches out and touches something that that something is something that it wants to eat? Versus, can you imagine that if these different pseudopods or arms are just reaching out and if they touched each other, right? How come the amoeba doesn't proceed to, like, in a very awkward way, try to eat itself, right? It doesn't have a brain. It doesn't think. It's all based upon these protein signals. And so it has to have receptor proteins on its surface that can actually detect when something is bound to these receptor proteins, that something is self, meaning it's part of the own amoeba, versus something else, like this, I don't know what this is, the scrap of something that it wants to consume, right? Self versus non-self recognition is a very important part of our adaptive immune system, right? And we will get into that quite a bit, especially when we get into the later parts of the semester, when we talk about different diseases that are caused by a failure of non-self recognition. So multicellular life, about 600 million years ago, you started getting diverse invertebrates, right? So insects like this fly here. And this is when, because you now have not just one cell, right, but if you have potentially hundreds of thousands, if not more cells, suddenly you realize you have to now have a system, right, of cells patrolling other cells and cells being able to call for backup, right? And so that's when the induced innate immune system began to evolve, right? And to this day, our induced innate immune system is the call for backup, right? Macrophages that send out cytokines to recruit things like neutrophils. But up until this point, organisms were still very short-lived, right? So even something like this fly might live as an adult fly for maybe a few days or a few weeks, right? So there wasn't a huge need for different species to develop an adaptive immune system that could anticipate something new. However, once we move on to the evolution of vertebrates, so the earliest bony fishes, right, all the way through mammals like us, we began to live longer. And as we live longer, that means we have an increased likelihood of encountering pathogens that our innate immune system did not come pre-programmed to deal with. So things like viruses or bacteria that are constantly evolving, with us, right? And end up causing infections that evolutionarily we're not sort of prepared to handle with our innate system. Therefore, we had to come up with a way to be able to adapt in order to recognize that something is in fact a threat, eliminate that threat, and importantly, remember that threat. Because if it didn't kill us the first time it infected us, right, we don't want it to be every time we're reinfected with it, we're gambling with potentially dying. Okay. So let's look at the conservation through evolution of certain aspects of our innate system, first of all, right? Because I think when I explained to you on the previous slide how the innate system is actually used by most life forms on Earth, and that the adaptive system came later, I don't want you to lose sight of the fact that the way our innate system works, all the stuff that we've learned about so far with complement proteins, macrophages, things like that, that's by and large how the innate immune system works in something like a fly, right? Here's an example. We studied in our cells, we have these special receptors on the surfaces of things like our immune cells, like macrophages, that can detect lipopolysaccharides that are shed by bacteria, right? These sort of hairy dogs, these bacteria, right, that are shedding their lipopolysaccharides, and our macrophages can pick up on that because they have toll-like receptors, or TLRs. And we said that the TLRs, they signal through the cytoplasm of the cell by kicking off a chain of different proteins talking with each other, represented in this cartoon here. Right. I don't want you to think that this somehow suggests that all these proteins somehow come together and form this, like, I don't know, solid thing here. No, this is representing that one protein ends up communicating with another, even though they're floating around inside of the cell. Right? But for simplicity's sake, it's just drawn as this one continuous kind of interaction. Ultimately, we learned that the stimulation of the toll-like receptor on the surface of the macrophage results in this very important transcription factor, NF-kappa B, that will then turn on cytokine genes in the macrophage. And that's how the macrophage starts releasing cytokine messages, right, to alert the rest of our immune system that something is happening. So remember, I said that it's called toll-like because the original toll receptor was identified in Drosophila. So if this is how this complicated series of protein interactions work in our bodies, in the TLR, you might think, oh, yeah, I bet it's a lot simpler in the very primitive fly, right? That sort of evolved hundreds of million years before we came along, right? So what does the original toll receptor pathway look like in a fly? It looks like this, right? It's the same thing, except it has different names because, Drosophila geneticists, they're kind of funny people. They have a much better sense of humor than most eukaryotic biologists do. Because when they identify different proteins that are important in, let's say, fly development or whatever, they give them funny names like cactus, and things like that, because it results, defects in that gene might result in different types of phenotypes. Also there's the hedgehog gene in Drosophila, and then the variation known as the sonic hedgehog gene, right? I'm not kidding you. right? They have a good sense of humor. But anyhow, each one of these proteins, though, is homologous. In other words, we as mammals, as people, have a version of that same gene that we depend upon as well. All right. So this is just to highlight the fact that much of our innate immune system is tried and true and tested over hundreds of millions of years of life evolving on this planet. So we still rely upon that. Right. So that then is really this upper part here, right? So all the lectures we've had so far has been talking about this upper part. And we said that the pros and cons of innate immunity are outlined here. We depend upon it like 99% of the time to get us through our day. Adaptive immunity is what we're going to talk about now, right? This is when the innate side of our system fails to successfully defeat the infection and defeat the pathogen, right? We have to now bring in the big guns, right? The heavy hitters, the expensive, specially trained SWAT team. And we said that they'll mount an overwhelming reaction. They'll somehow remember the pathogen for next time. But the cons of this are that it's going to take time for the SWAT team to get organized and mobilized. So from the time you're infected to when your innate system really couldn't handle it, and the SWAT team finally gets woken up, we're typically talking about a week to 2 weeks, right? And it's very expensive because when your adaptive immune system kicks in, right, you're going to use a lot of your body's resources in order to fight the infection. So let's go back to this cartoon then. We said that the innate effectors tend to be on this side of the cartoon on the right. The adaptive effectors tend to be on the left, right? They all come from the same hematopoietic stem cell in the bone marrow. And as that stem cell divides and differentiates, makes different decisions about what genes to turn on, what genes to leave off, or sets of genes to turn on and turn off. That's how they then become, right? All these different types of cells that make up your blood. So here you can appreciate, as I said before, the natural killer cells actually represent sort of the first cells that evolved towards this adaptive side. So this common lymphoid precursor at first, right, would just become this natural killer cell precursor and then become a natural killer cell, right? Later in evolution, this green common lymphoid precursor learned evolutionarily how to also become things like B cells and T cells. Right. So if you ignore the B cells and T cells, then you can actually see you don't have to draw this arrow sticking all the way to the left like this. It could be sort of more coming down this way to make it easier to appreciate that the natural killer cell still functions as an innate effector cell, right? Even though it represented sort of this next important step towards things like B cells and T cells. So let's talk about these. What are B cells and T cells? Hopefully, this is something that you're interested in, you've been waiting for, because I think many of you are aware of things like B cells, discussions about antibodies, and also T cells. Right? So let's get into our discussion about what these are. Let's start with the B cells. So B cells contain information for adaptive specificity. What do I mean by adaptive specificity? B cells are the things that are allowing our immune system to learn that something new that our innate effectors are not recognizing as being a threat are in fact threats. Right? So the way they do this is B cells make a special kind of receptor on their surface known as a B cell receptor. That's what these little Y-shaped things are. And they are attached to the surface of the B cell. Right? And these structures are what we call immunoglobulins, right? Or Igs. So these Igs, these really are those wanted posters that advertise the identity of a novel pathogen so that the rest of our immune system can learn from this and realize that it needs to look out for this. So in particular, let's say the wanted poster of bad clown here, right? So Igs are small proteins that do sort of 3 things. They uniquely identify an exotic threat. Right, so we often think of bad clown as being an exotic pathogen, something that doesn't come around every day, right? But maybe every few months or every few years you encounter something like bad clown, right? Antibodies, or sorry, immunoglobulins can also help by opsonizing the target. In other words, covering the target with immunoglobulins when complement alone cannot succeed. And importantly, immunoglobulins can help direct other effector cells, not only to recognize that it's a target, but help other effector cells focus their attention on attacking a target. So here you have a cartoon that shows a resting B cell. So its immunoglobulins are attached to its surface. Right? And so we call these immunoglobulins B cell receptors because they're still stuck to the surface there. Here, this B cell has encountered a bacteria. Right? And on the surface of the bacteria are different three-dimensional molecules. They could be, any kind of molecule, proteins, carbohydrates, lipids that happen to be the right three-dimensional shape to fit in the arms of the immunoglobulins, right? And that's how the immunoglobulin is kind of recognizing, or that becomes the identity of the wanted poster that these immunoglobulins are going to help advertise. Right? Importantly, three-dimensional surfaces that are the right shape to be detected by immunoglobulins are what we call antigens, right? So one way of thinking about it is an antigen is the part of a pathogen that actually is the three-dimensional surface that something from your immune system, like an immunoglobulin, is actually binding to and recognizing, right? What may happen at that point is the B cell will activate, and the activated B cell may become what we call a plasma cell. And that plasma cell then will start making tons and tons and tons of this specific immunoglobulin, but in a form that can be released. In other words, no longer a membrane-attached receptor form, but a free-floating protein, a soluble protein, meaning it dissolves in water so it can float through your blood. And this form of the immunoglobulin is what we call an antibody. So in other words, antibodies are immunoglobulins, right, in a form that's made by B cells that have turned into plasma cells that now can be released and flow through your blood. And those antibodies then can opsonize the target, and they can help with the recruitment of other effector cells. So let's take a closer look at these immunoglobulins. So immunoglobulins are these small glycoproteins. That means they're a combination of, a peptide or peptide chains with sugar groups on them. Each one of them is made by 4 polypeptide chains. Right? So you can see each one of these Y shapes here. There are these 2 big chains on the inside, starting from here. Here's one that goes all the way down to the bottom. Here's another one. So you have a pair of these big ones, right? And you have a pair of these smaller ones that are on the outside of each of the arms here. So 4 polypeptide chains in total. Right? And they come together to form this Y-shaped molecule. There are 5 variations or isotypes or classes of immunoglobulins, and they are IgG, M, D, A, and E. Okay? We also tend to use Greek letters to signify them. So gamma, mu, delta, alpha, and epsilon. All right. That shouldn't be too confusing because you just realize that, these are just the Greek letters of G, M, D, A, and E. Right? So one end of this Y-shaped, these 2 arms on this Y-shaped, recognizes antigen. The other end, the stem end at the bottom of the Y, binds to effector cells. Right? This would be the end that the immunoglobulin would be attaching to the surface of the B cell to start off with, as the B cell is making the receptor form of it. Right? That's the stem that anchors it to the surface. And then later, if the B cell turns into a plasma cell and makes a free-floating antibody version of this, these Ys are floating around, right? The red antigen binding arms would attach to the surface of the bacteria, and the other stem part of the Y would be sticking out. So other effector cells can benefit from that and latch on to the stem parts. Okay. So let's look at the structure a little bit more. So each one of these proteins is about 150 kilodaltons, right? That's the way we measure the molecular mass of proteins. There's 2 heavy chains. So here in this picture on the left, the 2 green ones are the 2 heavy chains, right? And we also call them H chains. And then 2 light chains, the smaller chains in yellow here. Okay? So these 4 polypeptide chains begin, when they're made, as simply 4 separately made small protein chains that then assemble together to form one Y shape. These chains are held together in this hinge region in the middle here by disulfide bonds. All right, so these are bonds that are typically resulting from 2 cysteine residues on the 2 chains, forming a disulfide bridge. And if you remember from Bio 275, that's a pretty strong way of influencing the proper overall shape of a complicated protein that's folded up, right, is disulfide bridges. And then these little blue things are the carbohydrate groups that are on it, which make this whole thing a glycoprotein, right? Importantly, another way of thinking about it is this middle panel here. Right? These red regions are the antigen binding sites. These regions are the N-terminal regions of these peptide chains. Right, so the N region would be here, and if you follow this heavy chain, you'd go all the way down the heavy chain. This would be the C-terminal region of or C-terminal end of that particular peptide chain. Right? So the N-terminal ends are also what we call the variable regions, or V regions, because these are the ones that are going to vary in shape from one immunoglobulin to another immunoglobulin, depending upon what the antigen is that they can detect. So if you think about it as a wanted poster, right, most of the structure of this Y shape is your generic poster, right? Like wanted and then, reward, whatever. That doesn't really vary that much from wanted poster to wanted poster. But the variable ends, the red ends here, would be the actual photo of bad clown in it. And that would change from wanted poster to wanted poster, right? So the blue regions are what we call the constant regions, because again, they stay more or less the same from one immunoglobulin molecule to the next. A third way of thinking about it is a very old-fashioned way, but it's important we mention about because the terms are still used today. Early researchers saw that if you were to kind of break up the immunoglobulin, that it would tend to fall into these typical pieces. You'd have the antigen binding fragments, or the FABs, so fragments that bind, antigen binding, right? And you have this fragment down here that would crystallize very easily. Right? So they called it the FC region. So that's what we also can think of as the stem region. All right, so different ways of thinking about these different parts, depending upon, where you're reading about it. They may refer to them with these different terms. So here's the 3-dimensional view of the structure. Right? So you can see here, the 2 heavy chains one is the blue, one is the yellow. So in the middle here, they'd be held together by disulfide bonds between the 2 heavy chains. And then on the outside of the arms, you have these 2 red regions, right, which are the light chains. So that's a nice 3-dimensional view, more realistic view of what it looks like. If we were to make it simplified, just so we can follow this a little easier, again, you can see in here, the red are the light chains, and then you have the blue and the yellow are the heavy chains held together in this Y shape, right? So it really is Y-shaped, okay? And each one of these is made of these separate globular blobby domains that are very conserved in terms of their overall shape. So we call these the different immunoglobulin-like domains. Right, so here's another illustration, again, showing the 3-dimensional space filling model of this. And you can see why immunoglobulins or antibodies are often represented as being Y-shaped. So let's take a closer look at what's happening here. All right. If we were to just zoom in on one of these light chains, so the light chain here is made up of 2 of these immunoglobulin-like domains, a variable globular domain, so V sub L for light chain variable, and a more constant region, so C sub L, so the light chain for the constant region, right? So just focusing on those 2, let's blow them up here, right? And here's the structure. So what you should be impressed with, first of all, is you can clearly see that there are 2 domains here, right? These 2 blobs. But importantly, this is all one peptide. So here's the amino terminus of the peptide, right? So if you followed this, it would be a chain of amino acid residues, and you just keep going, loop around, whatever. It's like a little maze. It's like a little game. Eventually, you'd go this way, and then it goes into here. You zigzag back and forth. Eventually, you'd end up at the C terminus of this peptide chain, right? So this is a chain of amino acids that then just folds up into these 2 domains. Let's see here. So importantly, these three sort of loop regions here, 1, 2, and 3, right, this is where the business end is of the antibody or the IG domain that recognizes unique three-dimensional shapes, right? Because this creates a three-dimensional shape that will match up with some sort of antigen. Most of these domains follows a pretty standard amino acid sequence. If you were to look from antibody to antibody to antibody to antibody, like if you took a sample of different ones, most of those antibodies would have similar structures in these regions here. But in these three loops, you have great diversity in amino acid sequence because these loops will be shaped radically differently from one antibody to the next. So if we were just to look at these loops here, so here again is that variable domain of the light chain of antibody arm, right? In red, they've highlighted those three loops that represent the regions of highest amino acid diversity. If you were to take this and unwind it and straighten it out, right, you would get something that looks like this on the left. So this bar on the bottom is you've unwound it. And so this might be the amino terminal end, the N terminal end over here, right, which would have been the start of it over here. But now you've kind of straightened it out, and you can walk through these different regions. The red ones then are the regions of amino acids that end up being the ones that are in these stretches. If you took a whole bunch of different antibodies, even from your own blood, one person, and you compared what are the amino acids that make up this region of these different antibodies, what you'd find is most of the yellow region doesn't vary that much from antibody to antibody. But once you get to these highly variable regions or hypervariable regions, the red ones, you see that there's great amino acid diversity, right, from antibody to antibody, meaning that in different antibodies, these three shapes would be very different. Keep in mind that this globular domain, right, is just the variable region of just the light chain over here. So it's pressed up against the variable region of the heavy chain. So the heavy chain is here. It kind of goes off to the left and would go down to the main stem part of the Y. So the antigen binding site of one arm of the Y shape is actually made of the hypervariable region of the light chain plus the hypervariable region of the heavy chain. So it's that combined 6 loops that creates a unique three-dimensional surface. So why is this important? Right. So one way of thinking about it is most of the structure of the immunoglobulin has to be kind of standardized. Think of it as puzzle pieces in a jigsaw puzzle, right? If you have a jigsaw puzzle here and you looked at 2 of the pieces that came out of it, you realize many of the qualities of these pieces are the same. They're standardized, right? Because they have to be the right size puzzle piece in order to even fit as part or belong as part of this puzzle. So you can think of that as the framework region of the immunoglobulins, right? So that's like the yellow part of the globulins that don't vary from immunoglobulin to immunoglobulin that much because the overall shape of the immunoglobulin has to be kind of standardized. These extra hypervariable regions, these hypervariable loops, represent these parts of the puzzle piece that actually give it its unique features so that the combination of these hypervariable regions means that this particular puzzle piece can only fit in one particular place in the puzzle, right? That's what gives it specificity. Right? Okay, so to illustrate this concept, right, using the puzzle pieces, I have here, let me stop the share for a moment. I have here a jigsaw puzzle, right? This is one of my kids' jigsaw puzzles. This is kind of a nice one because it's like a tropical coral reef, right? And, you know, and this puzzle is made up of 500 pieces, and you know, so the puzzle pieces are all kind of in here, right? And I think you all know that each piece has only one unique special place it'll fit in the puzzle, right? But I think we've all sort of done this before, where if you close it up, you kind of shake it around a bit, it around a little bit, right? What would you find? if you kind of look around inside of it and you start digging around in here, right, you might actually find examples of something like this, right? So here you have two pieces that came together, right, came together by chance, right, just by being mixed up in here. Now, these two pieces do not belong together. They are not meant to be together in the finished puzzle. But why do they actually come together? Oh, you see the light, you can see the little sunlight, right? Because they happen to be close enough fit, right? Not the intended perfect fit, but close enough, right? And so therefore, the binding is actually pretty weak. You just touch it and it falls apart. Right? So what am I getting at here? Right? I'm getting at the fact that if you have 500 pieces to work with, right, and you shake them around, there's a very good chance that a couple of them can come together by close enough fit. Right? All right, let me continue the sharing here. Oh my God, I can't tell which sharing I'm supposed to be running here. I don't think that's the right one. Desktop 2, PowerPoint, desktop. Maybe this one here. Oh no, this is this one here. Share this. Okay, and go back to this. Okay, so let me see here. Hopefully you guys can see this, that I actually didn't mess it up by turning off the share and turning it back on again. Okay, so can you guys just really quickly tell me, did it move on properly? So now it says novel antigen binding based on close enough fit?
Yes.
It's good. Okay, good. All right, thank you. So the point of this is, our bodies, right, are not psychic. Our immune system is not psychic. It cannot actually know what's going to come along, next week, next month, next year in terms of novel pathogens. So there's no way for our body to predict every antigen that we'll ever encounter. So the solution that evolutionarily we came up with is we produce millions of different variable domains in immunoglobulins at once. So your body is filled with B cells right now that are making hundreds of millions of variations of these immunoglobulins with hundreds of millions of variations of these hypervariable loops. And just like a mismatched puzzle pieces, what you're counting on is that if a pathogen does come in, right, that your innate system doesn't recognize, that you will already have some of these B cells with immunoglobulins that just by chance are close enough fit to start some recognition happening, right? So if you look here, let's imagine down here the puzzle pieces, the blue thing is the antigen, right? And then you have these two different V domains, right? This one variable domain, it's a close enough fit. It's not the right perfect intended fit, but it's close enough to fit. This other V domain, close enough fit over here, right? Importantly, the actual surfaces on the antigen that you can see here, where those V domains are interacting with, those are what we call the epitopes, right? So the epitopes are the actual three-dimensional feature on a three-dimensional molecule that a particular immunoglobulin can recognize. So of course, if we think about them as puzzle pieces, right, what we want to think about is the fact that the antigen is actually part or presented on the surface of a pathogen. And so these red and yellow puzzle pieces represent the variable regions of two immunoglobulins. Then in this case, you have two B cells, one that makes the red one that makes the yellow one. They are among hundreds of millions of B cells produced by your body today, right? But it just so happens that these two have close enough fit to actually work and be able to detect this pathogen. And so again, just as a reminder, the variable region, the hypervariable region, is this region here with those three different amino acid sequences in the loops. Okay, so one thing to keep in mind is that means your antibodies that you produce in response to an infection or a vaccine depends upon what B cells and immunoglobulins those B cells made in your body that day by chance. So the antibodies that you produce against the flu or the flu shot, right, are uniquely yours. So even if you and your brother or sister or roommate go and get the same vaccination, the antibodies that will ultimately be generated by your body to protect you are uniquely yours. All right. So this is the last slide. And all it is just to summarize, right? So B cells, when they first start off, they make B cell receptor. The first form that they make is the IgM form. You know, among the 5 types of Igs, the B cell first makes IgM, right? And if that IgM ends up being successful and binding to something like the bacteria, right, then the B cell will turn into a plasma cell and make the secreted form of an antibody, which may then change. It may no longer be the IgM form that's released. It may switch to a different form, such as IgG, as the secreted antibody. Right? And IgG is usually the type of antibody that medical professionals talk about. You know, when you talk about seeing if you have an antibody response or levels of antibody detected in your blood, that's because 70% of the Igs that are floating around in your blood as antibodies are the IgG form. Okay. All right, so that's the end of today. Next time, we'll learn how the B cells actually create such an amazing diversity of Igs, starting from the same genome, right? Because all of them are your cells, so they all have your genome in it. So how can you be making hundreds of millions of different variations of proteins every day in these B cells? We'll talk about that next time. Okay, we'll stop there.